Every movement that you make, from sprinting across a field to waving at a friend, depends on a complex architecture of your skeletal muscles. These voluntary systems don't just magically contract. They are highly organized mechanical frameworks that transfer force through a precise network of connective tissues, anchoring securely onto bones via tendons. While we only see the large, powerful movements on the outside, the actual structural blueprint is a beautifully organized hierarchy of layers packed tightly together. To understand how it works, we have to look past the surface and break down the physical anatomy from the macro level coverings down to the microscopic cables pulling the weight. Let's look at how this machine is built. First, picture your skeletal muscles as a giant biological Russian nesting doll, a system where every single layer you open simply reveals a smaller, perfectly organized version of itself wrapped inside. Let's have a look at one of the muscle bellies. Imagine cutting open a thick electrical cable. From the outside, it looks like a single solid wire, but inside you will find many smaller wires bundled together. Skeletal muscles follow the same principle. Each level is organized into smaller and more specialized structures, with every bundle contributing to the muscle's overall strength and function. The entire muscle belly is wrapped in a strong connective tissue called the epimysium. Think of it like the protective outer jacket of an electrical cable. It holds the muscle together, protects it from injury, and provides a pathway for blood vessels and nerves to enter the muscle. Without this supportive covering, the muscle would lose its shape and structural integrity during movement. Inside the epimysium, the muscle is divided into smaller bundles known as fascicles. Each fascicle is surrounded by another connective tissue layer called perimysium. Imagine a bouquet of flowers. Each smaller bunch is tied together before becoming a part of the entire bouquet. This perimysium organizes muscle fibers into functional groups while also carrying important nerves and blood vessels deeper into the muscles. Inside the fascicles are hundreds of individual muscle fibers, and each one is wrapped in a delicate connective tissue layer called endomysium. Although much thinner than the other layers, it provides support for every muscle fiber and helps deliver oxygen and nutrients. Now, let's zoom inside one muscle fiber. Instead of being empty, it's packed with thousands of long cylindrical structures called myofibrils. You can think of the muscle fiber as a pencil case filled with thousands of pencils. These myofibrils are the true contractile machinery of skeletal muscle, and when they shorten together, the entire muscle fiber shortens as well. Each myofibril is made up of repeating units called sarcomere, which are the smallest functional units of muscle contraction. Every sarcomere performs the same job, and because thousands of them contract simultaneously, they produce the force needed for movement. Inside every sarcomere are two major protein filaments, the thick filament called myosin and the thin filament called actin. These proteins are the real workers responsible for muscle contraction. Rather than shortening themselves, they slide past one another in a highly coordinated process. This is why the mechanism is called as the sliding filament theory. Actin is a protein that forms thin filaments. These filaments have active binding sites where myosin head attaches during muscle contraction. Myosin is a protein that forms thick filaments. It has head that can bind to actin and use energy from ATP to pull actin, causing the contraction of the muscle. It's like a rowing team sitting in their boat with paddles ready, but waiting for the referee signal before they begin rowing. The sarcomere segments span from one Z line to the other. They are dark zigzag lines that mark the official start and end of a single sarcomere. They hold the contracting units of the muscle together so they do not move apart during movement. Another important structure is the I band, also called as the isotropic band. This is the light colored isotropic region centered around the Z line. It contains only thin filaments that are actin and titin. Because there are no thick myosin filaments here, light can easily pass through it, giving it a lighter appearance. The anisotropic band, the dark band that spans the entire length of the thick filaments, the myosin. The A band contains both the thick myosin filaments and the overlapping ends of the thin filaments as well. This heavy protein density is what gives skeletal muscle its primary dark striations. H zone is a slightly lighter, central region within the A band where the myosin and actin filaments do not overlap. This zone contains only myosin filaments. Next is the M line. It is a dark line running directly down the exact middle of the sarcomere. It acts as a structural anchor for the heavy, thick myosin filaments. as it is formed by cross-connecting proteins that anchors and holds the thick myosin filaments in a perfectly aligned hexagonal array. In short, the skeletal muscle is a perfectly organized hierarchy. This hierarchy of skeletal muscle is essential because it solves a massive engineering problem. It multiplies microscopic movements into macroscopic force. Now that you have mastered the anatomy of the sarcomere, watch the next video on the sliding filament theory to see how exactly these bands and lines move to contract your muscles.